Okay, welcome back to the Classics of Immunology Journal Club. I hope you guys are enjoying all this. I certainly am. Every time I review one of these papers for this journal club, I learn all kinds of things that I didn't know before. You'd think that I would have read all these papers back in the day, and you know how it goes. You're busy, and so you basically skim them, and we also already know the data because you've heard about it in meetings 3 to 6 months before the paper actually comes out. And so a lot of times you never read the papers carefully, and therefore you don't really know whether what everybody concluded was really true or not. So anyway, this one's a particularly complicated paper. But before we get started, don't forget to like the video and click on the bell and get notified for more. Then scroll down on the YouTube site and check out my website. And you can find most of the classic papers there under the writing tab. And also check out the social media. I've been tweeting a fair amount, keeping up with COVID-19, you know. The paper that I'm going to review today is entitled The Cloning of Complementary DNA Encoding T-Cell Replacing Factor. And the authors, there are multiple authors, but I just want to highlight Tatsuo Kinashi, Eva Severinsen, Pascala Sideras, Kiyoshi Takatsu, and Tsuku Honjo. And so this is another collaboration between, now we got 3 labs involved, Severinsen's from the Karolinska, Kiyoshi Takatsu from Kumamoto University, which is on the southernmost island, Kyushu, and Tsuku Honjo, who had now emigrated from Osaka University to Kyoto University. So this paper was published in Nature, another Nature paper, in November of 1986. And so in order to really get into this paper in terms of what these people are saying that they have done, it's in the whole history of immunology is important here. And I just want to recall your memory of the first definition of T-cell replacing factor that was by Anneliese Schimpel and Eberhard Wecker, published in 1972 in a paper in Nature New Biology that I reviewed here on the Journal Club. And I went back and reviewed that video. And so you might want to do that too before you get into this particular video. It's very interesting. And because what Schimpel and Wecker, to really boil it down, is that they did mixed lymphocyte cultures between thymocytes from 2 different strains of mice and took the supernatants and added them to the Michel-Dutton plaque assay method. For the Michel-Dutton assay was to take mouse spleen cells, mix them together with sheep red blood cells as the antigen, culture for 4 days, and then look for plaque-forming cells for IgM. And so that was the Michel-Dutton assay, which was very important for the B-cell people. And they found that if they, it had been found in 1970 by a couple of 3 groups, that if you took the T cells out of those spleen cells in the Michel-Dutton system, that you didn't get any plaques formed. And so what Schimpel and Wecker were saying is that you could get a soluble product from T cells that were released via the activation of the mixed lymphocyte culture that would replace T cells. You don't need any of these T cells anymore. So that's the background on that. Between 1972 and 1986, Kiyoshi Takatsu was very interested in trying to define the molecular basis for this T cell replacing factor. And the assays that were developed over the course of, like 14 years or so, had morphed from the Michel-Dutton system to using cell lines, B cell lines. And Susan Swain and Dick Dutton had published in 1982, a decade later, after the Schimpel and Wecker paper, that they had a cell line that was called leukemia lymphoma cell line from a mouse that was called BCL1. And that became the target cell line for at least 2 activities. One was the proliferation of those cells under the influence of different factors and so forth. And the idea is that the T cell replacing factor would cause those cells to proliferate more so than they would otherwise. But in addition, what Kiyoshi had worked up is he was interested in more, not quite so much in the B cell growth factor, which is what Swain and Dutton had called it. You recall now, this gets complicated, you recall that Bill Paul also had a B cell growth factor that he was talking about. And he assayed his factor derived from T cells on anti-mu activated splenic B cells. And so that became essentially B cell growth factor 1. And this, the factor that caused the BCL1 cell line to proliferate was BCGF2. And that became one of the assays then that for T cell growth factor. But the other assay that Kiyoshi had worked out was a plaque-forming cells for IgM synthesis from this particular cell line. Kiyoshi had these assays going, and he was trying to purify the active component. And of course, as you know, that wasn't an easy thing to do back in those days for all kinds of reasons. But it turns out that after Hanjo's group had made a cloned a cDNA library that had the activity that we reviewed last week in terms of interleukin-4. It turns out that Kiyoshi's assays would also detect the same cell line that had been used to construct the cDNA assay by Ava Severinsen and Pascalis Sideros. The interesting thing is that as a consequence of last week of me reviewing that paper, the interleukin-4 paper, I contacted Pascalis Sideros by email. So good to do these electronic communications these days. And it turns out he's at the time he was the postdoc that was really doing the experiments, the biological experiments that were in all of these different assays. He was Greek. He went back to Athens and has had a long career. And he answered my email out of the cyberspace from La Jolla, California. And so we've traded war stories back and forth. And he basically told me that the assays, that the screening assays for the cDNA clones and so forth that he did were he could tell by looking at the cells as to whether or not there was a positive or not. And I said, is that because they changed from small round cells to elongated cells? And he said, absolutely. That's exactly what happens. And of course, I knew that happened back in the day because I saw that early with IL-2. And I guess it all has to do with the different signal transduction pathways and what is activated. But actin and myosin get activated in these cells that makes them have all these funny shapes. So that's just a little bit of the background. The thing is that when interleukin-4 was tested in the assays, interleukin-4 now it was defined as it would cause an induction of IgG1 and would also cause the cells to proliferate. And these were the I'm pretty sure the LPS activated splenocytes. So anyway, so they had these assays, and they had. The other thing that had happened was that Severinsen and Sideros had a cell line. It was a T cell line, IL-2 dependent T cell line, designated 2.19, that would spontaneously release factors that would seem to be help scoring in these assays. And the assays, you had to have a scorecard. I mean, there were so many. Each group, there was about at least a half a dozen at this particular point in time that were they're all competing with one another. This is the early days of the interleukin revolution, as I call it, because that was the beginning, the IL-1, IL-2, 3, 4, and so forth, 5, and so forth. And now we're up to more than 40 years later, or so, 30 years later. And so they were very competitive at the time, and everybody had a different assay system, and they named their factor based on the assay system, and they couldn't be sure that the other guy's factor was the same thing or different from the factor that they were working with because they were using different assays. The obvious solution to all this was to purify the molecule, find the molecule that was really causing these cells to behave as they were. And so that was sort of the quest that these people found themselves on. And so in this paper that came out in November of 1986, I should mention that in January of 1986, there was a paper that came out in PNAS from a group in London. It used the assay cells that Kiyoshi had used, these BCL1 cells that were originally derived are used by Susan Swain and Dutton. And remember now that caused those cells to proliferate, and that was BCGF2. The Mill Hill group in January 1986 in PNAS published a paper without the molecular characterization, but with indirect evidence that this factor that they were dealing with, and they were trying to find factors that would cause bone marrow cells to proliferate and differentiate into different kinds of white blood cells. And there was one particular white blood cell that was called an eosinophil because it had big granules in it that were chock full of very basic proteins that would take up acid dyes. And that was where the eosinophil. Eosinophils are only comprised anywhere from 2 to 5% of the total polymorphonuclear leukocyte population. And so, but the interesting thing about eosinophils is that they were often present in severe allergic phenomenon, severe asthma. And they were also quite frequently found in patients who were infected with parasites. And so really what these eosinophils did, or how they did what they did and so forth, this is what the group in Mill Hill were looking at. And they decided to call their factor interleukin 4. Well, and then subsequently Gahanjo and Severinsen and Sideros, did their work that came out in Nature, I think April or May of 1986. So this whole 1986 was a confusing year for factors. But this figures into the background of this particular paper. So the materials and methods, which again are found in the figure caption in the font that is like -4. So I had to use a magnifying glass to really make sure I knew what I was thinking about, I was reading. The BCL1 cells, when they were cultured together for 2 days with the factor supernatant, and using the protein A plaque assay, you could demonstrate IgM positive plaques. That was number one assay. And the second assay was same thing. You start with the BCL1 cells, you give them the soup or the factor, and they will then proliferate more. And that was Swain and Dutton's assay for BCGF2. Also after 2 days, you just threw in tritiated thymidine and waited a while and then harvested them and counted the counts. I forgot to say that Kiyoshi Takatsu had a hybridoma, T-cell hybridoma cell line that was called B151. Had to be B, and it couldn't have been C. So because it's confusing, why don't they call it a T151? Anyway, that becomes important because that was the source of the factors that were posited in Takatsu's assays. They also had the Severinsen and Sideros cell line 2.19 that I talked about before. And it turns out that these soups from these two sources of T cells were positive in Kiyoshi Takatsu's assays. That's important because, and this is what Pascalis Sideros informed me, which I should have picked up on myself, was because the cDNA libraries that they used to clone out interleukin 4 was from the 2.19 cell line. Well, when they were tested in these other assays that Kiyoshi Takatsu had, they were also positive there. So they had already done the work. They didn't have to reclone anything. So they basically took their cDNA clones that they had stashed away in the lab, and they started working on those to see whether or not they could identify whatever T cell replacing factor was. And so that's it. Now the results are just two results, and I'm going to do the screen sharing now. This is table 1, and this really has the meat of the data. So let me make sure I have all my notes here. So the first thing to look at is the B151 T cell hybridoma supernatants percent. IgM plaque-forming cells per culture of 1,800. Thymidine incorporation 7,000 CPMs per minute. So that was the positive control. And that was done at different dilutions, and it dilutes out. Then here's supernatants from the 2.19 T cell line. Again, using less, only 1%, and then titering it out. Positive in the IgM plaque-forming assay and positive in the thymidine incorporation assay. Here's a negative control, HeLa cells. Those are the Helen Lane cells that have gotten so much press these days. Let me see, yeah, plaque-forming cells, 200, and very low thymidine incorporation. And here's the cDNA derived protein, the recombinant interleukin-4 protein, translated by the phagocytes. And now this is important because this is interleukin-4. The IgM plaque-forming cell culture was negative, but the BCGF culture was positive. By comparison, the B151 TRF was positive in both assays, which meant, though, that the BCGF part of the whole thing couldn't be used, since they were both positive here, couldn't be used to discriminate. So they had their cDNA library. They digested the library and with different restriction enzymes, and they found positives. So they took the whole library. They did 18 pools with 3,000 clones in each one, and they found one pool out of those 18 that was positive. They then subcloned, and they ultimately ended up with one clone that had a large insert of at least greater than 1 kilobase, and that clone was number 23. So you can see, if you look at number 23 here, it's positive in the IgM plaque assay, and it's positive in the thymidine incorporation assay. And then there's negative and positive controls for this whole business here, but that's it. They used that clone, number 23, and they did their MoBio parts of the experiments. And this is the summary of those data. This is the cDNA sequence on the bottom row of each of these, and the top is the predicted amino acid sequence. The cDNA had 1,500 base pairs. The open reading frame had coded for 133 amino acids, which is about the size of IL-2. There was a 21 amino acid hydrophobic signal peptide, so that fit well with a secreted protein. The secreted protein had an estimated molecular size of 12,300 Daltons. And that when they went onto their databases to look for homologies with other genes, they found that there was some homology with interleukin-3, mouse interleukin-3 in 2 different areas, and some homologies with mouse gamma interferon, and also homologies with GM-CSF. So they were pretty sure that they had cloned out a whole different thing. And of course, there were no homologies between other interleukins and so forth that had been molecularly characterized, so that there was no homology with interleukin 1, 2, 3. Well, there was 3 there. Interleukin 4, and so they were quite happy with this. They submitted it to Nature, and it was published in November. And simultaneously, as I'll tell you in a little bit, there was another paper out on another lymphokine that was published back to back with this particular paper. And they, in their discussion to this paper, they decided that we don't need to screen share anymore. So in the discussion of this paper, they say that of all the interleukins that had their primary structure determined, this was the fifth in the series, and so they proposed to name this particular interleukin 5. Now then, back to the Mill Hill publication that was out in January. Colin Sanderson and Jerry Klaus from Mill Hill, and Ann O'Gara was on that paper too. And they had been looking for this eosinophil differentiation factor and using bone marrow as their target cells and so forth. And they had not purified or cloned the cDNA for this particular factor they were working on, but it scored positive in the BCL1 BCGF2 assay. And it turns out, as time went on, after all these papers were published, was that this was the human counterpart of murine IL-5. And that became important because that was the human so-called TRF, T-cell replacing factor. But in the end, as time has gone on, I just want to mention the fact that the B-cell part of this that Kiyoshi Takatsu had worked so hard on and so forth, in humans, it really didn't have a lot of B-cell activity. The primary activity of the interleukin 5, that this thing was called essentially, was the eosinophil differentiation factor activity. So after all this was said and done, the quest to find out how T cells helped B cells essentially boiled down to interleukin 1, interleukin 2 had B cell activity, and interleukin 4 had B cell activity. And there was one more that I'm going to talk about next week, which was interleukin 6. But that still did not really complete the picture until 5 years more. And I think I mentioned this last week when CD40 had been discovered and CD40 ligand on the surface of T cells. And so there's more to come. And thanks for watching. Don't forget to like the video and click on the bell and check out my website and my social media sites. Thanks for listening. Bye-bye.